Electromyogram is a tool that we use to look at the current that causes by muscle contraction. Muscle contraction begins with ion flow moving across the muscle cell membrane, and it causes the depolarization of the muscle fiber or muscle cell. The flow of electrically charged ions causes electrical current. And if you use electrodes on the surface of the skin, we can detect this current and visualizing it using a computer program. And this is called electromyogram or EMG. Here, it's showing EMG trace, which is the subject has flexed his arm. The forearm is flexed by contracting, making a fist. And throughout these experiments, these flexors are constantly working and contracting. We have another way of doing this experiment, which is called motor unit recruitment. In this one, the subject is going to progressively give us a stronger grip force. And what happens when you give stronger grip force, more motor units are activated, more muscle fibers are contracted. So successively, you have higher EMG response. Here in the y-axis, we have the amplitude of response in millivolt. Here is our time in second. And as we are recruiting more motor units, we get a stronger and stronger response because more of motor units are activated. The amplitude of response is higher and higher. Here we're looking at neuroendocrine system. Neuroendocrine system is important for the homeostasis control of the body. Basically, body will be in balance. We need endocrine system. Endocrine system has two major components: nervous system and endocrine system. Nervous system response is rapid and transient. You have two subunits in the nervous system: central nervous system and peripheral nervous system. Central nervous system is made of brain and spinal cord. This is the commander of the nervous system. And peripheral nervous system is all the supporting nerves going to different part of the body. And the neuroglia, these are the supporting tissue. And for example, he's showing nerves, here's showing nerves to lower limb, upper limb, face, and so on and so forth. These are peripheral nervous system that's supporting the command going to the central nervous system and coming back to the affected organ or affected tissue. Endocrine system has this response, which is with hormones. So the response are slow, but it's long lasting. Here we're looking at a few endocrine glands, thyroid glands. On the anterior view, you have thyroid. On the posterior view of thyroid, you have these four little small glands. They are kind of like sphere shape. In the posterior end of thyroid gland, we don't see it here. They are called parathyroid gland. They're important in regulating the calcium. So thyroid gland, it's kind of like a butterfly shaped, and it's in the anterior neck. It's located here. And in the posterior end of the thyroid, you see the four small glands located. They're called parathyroid. You have adrenal glands above the kidneys, adrenal glands. Pancreas. In the pancreas, you have a group of cells. They're called islets of pancreas, or islets of Langerhans or pancreatic islets, those are endocrine in function. And endocrine in function means they are secreting into the bloodstream as opposed to exocrine like skin, like a sweat glands, which is releasing its content to the surface, not through the bloodstream. You have ovaries in female and testes in male. Those are also part of endocrine system. Here is showing histology view of the pancreas, and this group of cells, islets of Langerhans, these group of cells are endocrine in function, and this is a histology section of pancreas. And this cluster here is your pancreatic islet or islets of Langerhans. Here we see thymus, also part of endocrine system. We have here you are looking at pituitary gland, which really is made of two glands. One part is the glandular structure, which is called anterior pituitary, and this part is called posterior pituitary, which is kind of like nervous tissue structure. This is more in the anterior end of brain. In the posterior end of brain, you have pineal gland, which we're going to see in the next lab where it's located, both pituitary gland and pineal gland. Here we are looking at the neuron anatomy that we started talking about it in the lab 5. Here we go deeper. We look at a neuron. This is a multipolar neuron. It has multi-poles, okay? These are called dendrites. The cell body is called soma. The nucleus is in the center. This point of connection to this long extension, which is called axon, this is the axon. This part is called axon hillock. And you have Schwann cells that deposit this myelin sheet around the axon, and where you don't have myelin sheet, it's called nodes of Ranvier. These are important because now you can have the signals jumping through these and get to the target tissue much faster. You have axon terminal here, and then here we're going to go and connect with the tissue that is talking to this neuron. You have neuroglia, which is non-conductive tissues like Schwann cells that deposit this fat substance, this myelin sheets around the axon to protect it and to facilitate the speed of the transmission. Is called Schwann cells. This is one type of neuroglia. Here, we're going to go to peripheral nervous system. You have 12 pairs of cranial nerves. These are part of peripheral nervous system, and 31 pairs of spinal nerves. Cranial nerves in function, they are either sensory or afferent. What does that mean? That means they carry impulses towards central nervous system, or they're motor in function or afferent. They carry impulses away from central nervous system, or they are mixed, basically more both sensory and motor. They are in one nerve bundles. These cranial nerves, they are located inferiorly. When you look at the brain, you have to go inferior of the brain, and they are numbered in order anterior to posterior. Spinal nerves, there are 31 pairs of them. They're all mixed in function, so they have the bundles of both sensory nerve in one nerve bundle. They are numbered based on the area, name and numbered based on the area they are associated with that vertebrae. For example, cervical, thoracic, lumbar, sacral. Here we're looking at inferior view of the brain. This is the anterior end of the brain. This is the posterior end of the brain. This part of the brain in white is called cerebrum. This in pink is called cerebellum, and this is the spinal cord in white right here. Now I'm concentrating on the cranial nerves in dark pink. These are olfactory nerves, one, and you have them in pairs on both sides. Olfactory nerve 1, optic nerve 2, oculomotor nerve 3, trochlear nerve 4, trigeminal nerve 5, abducens nerve 6, facial nerve 7, auditory nerve 8, glossopharyngeal nerve 9, and then we go to vagus nerve 10, spinal accessory nerve 11, and then we're going to kind of come back to the top. We have hypoglossal nerve 12, these two, hypoglossal nerve 12. So they're in pairs on both right side and left side. This is a mnemonic device for you to learn the cranial nerves. You have olfactory nerve 1, optic nerve 2, oculomotor nerve 3, trochlear nerve 4, trigeminal nerve 5, abducens nerve 6, facial nerve 7, auditory nerve 8, glossopharyngeal nerve 9, vagus nerve 10, spinal accessory nerve 11, and hypoglossal nerve 12. So we're calling it, the mnemonic devices on old Olympus's towering top, a fin and German viewed some hops. On old Olympus's towering top, a fin and German viewed some hops. That's how you remember the orders of them, at least. And then as far as the functions of these cranial nerves goes, you call it sensory, sensory, motor, motor, both sensory and motor, motor, both sensory and motor, sensory, both sensory and motor, both sensory and motor, motor and motor. Some say marry money, but my brother says big brains matter more. Here we're looking at the spinal nerves. They are, as far as their function goes, they are mixed. So they are both sensory and motor in function. You have 8 pairs of cervical nerves. They are called cervical spinal nerves C1 to C8, T1 to T12 for thoracic nerves, L1 to L5 for lumbar, S1 to S5 to sacral, coccygeal, you have C0, 1 pair. So you have cervical nerves, thoracic nerves, lumbar, sacral, and coccygeal. Here we're looking at plexes. Plexes are these collection of, they're kind of like braiding, collection of nerve that they innervate a specific region of the body. For example, you have cervical plexes. They innervate head, neck, and diaphragm. Brachial plexes, they innervate the upper limb. Lumbar innervates the anterior thigh, and sacral innervates buttocks, lower limb, and pelvis. So it's marked for you here. Cervical plexes is C1 to C4. C1 to C4, so part of the cervical nerves. You have brachial plexes, C5 to T1. You have lumbar plexes, which is L1 to L4, and sacral plexes, which is L4 to S4. These are kind of like braided, as you can see, they're kind of like intertangled. When we look at the human brain, this is a lateral view. We have cerebrum, cerebellum, and spinal cord here. Okay, this is all central nervous system. Human brain is made of different lobes. We have frontal lobe, parietal lobe, occipital lobe, temporal lobe. This is cerebellum. You have between cerebellum and cerebrum, you have these fissures, these deep grooves. They have different names. For example, this one between cerebrum and cerebellum is called transverse fissure. Between temporal lobe and parietal frontal lobe, this fissure, this deep groove, is called lateral fissure. And this one is not a very deep groove. That's why it's called sulcus and not fissure. Between frontal lobe and parietal lobe, this is called central sulcus. And you have the shallow grooves, they're called sulci, and you have it all across the brain, sulci, and the meaty part in between the sulci, these raised areas, they're called gyri, sulci and gyri. We're looking at the sagittal section of human brain. Again, this part is our cerebrum, and this is our cerebellum, and transverse fissure here is also very obvious between the two parts of the brain. Inside the cerebellum, you see arbor vitae. It's kind of like tree of life. They look like tree branches. Cerebellum with arbor vitae. Here, the connection between the two brain hemispheres, we have corpus callosum, we have septum pellucidum, which is a very thin sheet, and if I poke through that sheet, it takes me to this space which is called lateral ventricle. Lateral ventricle. It's below this septum pellucidum. And then you have fornix right here. This is thalamus. You have hypothalamus. You have pituitary gland here. This is optic chiasma. It's a cross that brings the information from right eye to the left occipital lobe and from left eye to the right occipital lobe. Optic chiasma, pituitary gland. This structure is called mammillary body. Mammillary body. You have brainstem. You have medulla oblongata. Pons is part of brainstem right here. You have medulla oblongata here. Here we are looking at these spaces that cerebrospinal fluid passes through. This space is called fourth ventricle, and fourth ventricle is connected to third ventricle through this opening, this passageway, which is called cerebral aqueduct. And we already talked about lateral ventricles here, which if I poked through the septum pellucidum, it would take me to lateral ventricle. It's marking us the gyri and also sulci. And this one's parietal lobes, occipital lobe, and frontal lobe. We're looking at the transverse section of brain. And again, this part is our cerebrum. This is in green is our cerebellum with arbor vitae, right? And here I'm looking at corpus callosum again, septum pellucidum, and fornix. This gooey structure is choroid plexus. Here I have superior colliculi number 31, inferior colliculi number 32. Collectively, there are four structures. They're called corpora quadrigemina. Here I'm looking at pituitary gland. This is pons. This is mammillary body. This is optic chiasma, hypothalamus, and thalamus. Here we are looking at human brain ventricles. Again, ventricles are a complex series of spaces within the hemisphere of brain, which produce and also house cerebrospinal fluid. So we're looking at lateral ventricle here, below septum pellucidum. We have third ventricle around the thalamus. you have cerebral aqueduct. Aqueduct of Sylvius is also cerebral aqueduct, which is connecting the third ventricle to fourth ventricle. And this is your cerebellum, and this is your cerebrum, and this is your spinal cord. Dura mater, this one I have to explain more about these cerebral meninges. These are protective layers that cover the brain and the spinal cord. And in the brain, in the cerebrum meninges, this protective layer gives more modification in two areas. One is called falx cerebri, which penetrates longitudinal fissure, which is between the two brain hemispheres, and tentorium cerebelli, it penetrates the transverse fissure, which is between the cerebrum and cerebellum. So these are the two modifications that you have in this protective layer of connective tissue, which is called cerebral meninges. Falx cerebri penetrates longitudinal fissure between the two brain hemispheres. Tentorium cerebelli penetrates the transverse fissure that separates cerebrum from cerebellum. Here we're looking more at a cross-section of the spinal cord to look at the meninges in the spinal cord. This is the anterior end. This is the posterior end. How do I know this is posterior? Because spinous process of vertebrae is a posterior feature. Body of vertebrae is an anterior feature. And I'm looking at it superiorly. So here I have, if I'm going from outside in, I have this layer is called dura mater. These are the meninges that we're looking at. Dura mater, below it there's a space, it's called subdural space. Then we get to the arachnoid mater. Then below arachnoid mater, I have subarachnoid space. And then subarachnoid space takes us to pia mater, which is like a saran wrap covering, this is a cross-section of spinal cord, covering the spinal cord. Your spinal cord also has white matter and gray matter. The gray matter, which is in green here, has three parts to it: ventral gray horn, lateral gray horn, and dorsal gray horn. Here, this is the dorsal root. This is the ventral root. And I know this is dorsal root because dorsal root ganglion, which is the cell body of motor neurons, are located in dorsal root ganglion, is a dorsal feature. Okay, here we have sheep brain, and we're going to look at it as a whole, and then we're going to dissect it. So here I have cerebrum, I have cerebellum and spinal cord. Here you have frontal lobe, parietal lobe, temporal lobes on both sides, and occipital lobes. Okay, and then we are going to look at the sulci and gyri, longitudinal fissure between the two brain hemispheres, and transverse fissure between cerebrum and cerebellum. Here we're looking at the cross-section of sheep brain. This is a dissected sheep brain. So we're going to name some structures here and some here. Okay, so this GUI structure here in black is choroid plexus. Choroid plexus, cerebrospinal fluid is made here, choroid plexus. Here you see a little bit of septum pellucidum. I think we could see it a little bit better on the other side. However, if I remove the septum pellucidum, here is showing us the lateral ventricle. Okay, so we have septum pellucidum here. Optic chiasma is showing here. Mammillary body is showing here. This passageway right here is called cerebral aqueduct between the fourth ventricle and third ventricle, which is around the thalamus here, cerebral aqueduct. You have pineal glands showing here. transverse fissure between cerebrum and cerebellum. You have superior colliculus, inferior colliculus combined together. They're called corpora quadrigemina. If we look at this section, we are seeing spinal cord on both sides. We see medulla oblongata in front of cerebellum. We see arbor vitae here. We see fourth ventricle, and we see cerebral aqueduct, this passageway. Again, inferior colliculi, superior colliculi shows thalamus, hypothalamus. Here we have optic chiasma again. We have corpus callosum. This one, septum pellucidum, is not shown. It's open and it's taking us to the lateral ventricle. And here you have fornix. Oh, and then pons here. If you look at the eye model, this is human eye model, we have frontal sinus here. There are muscles of the eye. If they're going straight, we call them rectus. If it's on the superior end, we call them superior rectus. Superior oblique if they're going diagonally, inferior oblique. And we have inferior rectus, which is this one right here. And then you have lateral rectus and medial rectus. The way we know which one's lateral, which one's medial, if you look at the posterior end of the eye, there is optic nerve 2, but optic nerve 2 is not located exactly in the middle of the sphere. It's more towards the midline, so it's more medially. That's how we know where the midline is. That would be our medial rectus. The other side would be our lateral rectus. This is showing lacrimal gland. This white sphere that keeps the eye intact, the shape of the eye intact, is called the sclera. And cornea is this clear covering in front of the eye. Cornea, sclera, frontal sinus, superior oblique, going diagonally, inferior oblique in the bottom, superior rectus, inferior rectus, lateral rectus, and medial rectus is on the other side. Here we're looking at muscle of the eye. Again, on the top, you see superior rectus. And here we're seeing also lacrimal gland. Here we have cornea, we have sclera, we have iris, the colored part of the eye, and pupil. This is the duct, the canaliculi for the lacrimal gland, lacrimal canaliculi. Here we're looking, again, muscle of the eye is on the top, but this is really, we're going to look at the eye itself. This is the superior rectus again that we're looking at. Sclera here, cornea, the clear part of this covering. Iris is the color part of the eye, and pupil is the opening that light goes through. Here again, we're looking at the muscle of the eye. Superior rectus is shown, lateral rectus is shown, and here we see frontal sinus, sphenoid sinus, and this is the optic nerve 2. This one is, we're looking at pupil, iris, cornea is the colored part, and cover, the clear cover, the sclera. And here I can see inferior rectus again, inferior oblique again, and we have retina, and below retina, we have choroid. Choroid is that kind of like a black box inside the eye that doesn't allow the reflection of the light going through, and retina is where all your sensory receptor for the eye is located. When we look at the eye, if we make a cross section, coronal section, you have the anterior part that there's a space, it's called anterior cavity. This is in front of the lens of the eye, and in the back of the eye, behind the lens, is called posterior cavity. In the posterior cavity, we can find, this is the retina that we can see in the posterior cavity. We can find the blind spot, which is called optic disc, and fovea centralis, which is the most focal point. And if I put my probe inside this fovea centralis is like a pit. It goes about 1 millimeter deep. Here you have the most number of sensory receptors. And the optic disc is your blind spot, and that's where all the nerve ending ends to merge to optic nerve 2 and connects with the brain. This is cow eye dissection, and this is the anterior. When you make that coronal section, this is the anterior portion of it. You see the cornea, the clear part of the eye, and sclera, the white sphere around the cornea. Here, again, cornea and sclera. Here it shows pupil, iris around the pupil, and this space below this opening, below this hole and in front of the lens is called anterior cavity. Then you have your lens, which looks like kind of like a bath bead. And then this vitreous humor is this fluid that keeps the shape of the eye together. It's kind of like it's not fluid, it's like a jelly substance, vitreous humor. You have in the posterior cavity, you can see retina in white. And if I swirl retina around, it gets stuck somewhere, which is at optic disc. And when you look at the fovea centralis with the pit, it's a pit with your probe. You can actually go inside this pit. That's how you know you are in fovea centralis. Choroid is this kind of like blue and black structure. These animals, they have night vision. That's why their choroid is not black like humans are. And then you are seeing optic nerve 2 here as well at the back of the eye, in the posterior end of the eye. We're going to look at the ear structure. You have the external ear, which is the auricle or pinna, the external auditory canal. Then you have the tympanic membrane. You go to the middle ear, which is made of auditory ossicles. There are 3 small bones that make inside this air-filled structure inside the middle ear. Then from there, you go to the fluid-filled structure. This is your inner ear structures. You have semicircular canal, vestibule. These are important for your balance and stability and the sense of place in the sense of being in the place and in your surrounding. And then you have cochlea, and inside the cochlea you have organ of Corti, which is important for translating the sense of hearing here in organ of Corti. And this is auditory tube or eustachian tube here. That's another model, ear model, pinnae, external auditory canal, tympanic membrane. Here you can see the middle ear bones. This one's called malleus, incus, and stapes. Malleus, incus, and stapes. Here you have semicircular canal, vestibule, and cochlea. So if I took a cross section of cochlea and I went inside and I magnified it under the microscope, I would see the organ of Corti. I have fluid-filled structure here, fluid-filled structure here, and only this part is called organ of Corti. This is tectorial membrane. These are hair cells, and this is basilar membrane. Basically, when the sound wave come through here, they vibrate the tympanic membrane. In turn, they vibrate these ear ossicles and amplify the sound. The stapes is going to vibrate the fluid-filled structure here. It bends the tectorial membrane. It bends the hair cells, which have these hair-like projections, and action potential occurs through the basilar membrane, through the auditory nerve. You send the signal to the brain, and you can hear that sound. Here we're looking at the tongue model. These are papilla, each of these, and this is the epithelium. If I went inside in the microscopic structures, we could see the taste buds. This is the dorsal surface where the epithelium is. The papilla are these bumps, and taste buds, which you can only view microscopically, are these little structures. These are your taste buds. Here we're looking at two types of papilla, circumvallate papilla, mostly in the posterior end, in the dorsal end. These are circumvallate papilla, and fungiform papilla is all across your tongue. Most of the tongue has fungiform papilla. The circulatory system is cardiovascular, so we start with the human heart, and this is showing the anterior view of the human heart. When we're looking at it from the frontal view, from the anterior view, we have this groove that is called anterior interventricular sulcus. This is going in the middle of the heart, and it's between the two ventricles, these two spaces, these two chambers of the heart, and it's in the anterior view. So it's called anterior interventricular sulcus. And we have on the right side of the model, we have this chamber, which is called right ventricle. Above it, this chamber is called right auricle. We have left auricle, left ventricle. These are the four chambers of the heart. We have this blood vessel is called pulmonary trunk. Pulmonary trunk branches into left pulmonary artery and right pulmonary artery, which we don't see here in this picture. You have left pulmonary veins and right pulmonary veins, which are not seen in this picture. We have superior vena cava that brings all the deoxygenated or less oxygenated blood back into the heart through superior vena cava. And then superior vena cava branches into left brachiocephalic vein and right brachiocephalic vein. You have this blue blood vessel is called great cardiac vein. It actually sits in the anterior interventricular sulcus, and you have anterior interventricular artery here. We also see here, these are called coronary arteries and veins. These are the blood vessels that feed the heart itself. This is called, in red, it's called right coronary artery. Right coronary artery. This little guy here is left coronary artery, which is divided into circumflex artery, goes this way, and anterior interventricular artery. When we look at aorta, this is the biggest artery in your, biggest and longest artery in your body. You are looking at sections of it. If it's going up, we call it ascending aorta. When it's in the arch, aortic arch, and then we're going to go down, it's called descending aorta. If it's above the diaphragm, we call it thoracic aorta. If it's below diaphragm, we call it abdominal aorta. Aorta is further subdivided into brachiocephalic artery on the right side, left common carotid artery, and left subclavian artery. So this is the pulmonary trunk, and we are looking at this chamber inside the auricles, right atrium, left atrium, right ventricle, and left ventricle. This is an anterior view of a coronal section. So we kind of made a coronal section and we opened the heart and we're looking inside the heart. The valve between the right atrium and right ventricle is called tricuspid bicuspid valve. The valve between left atrium and left ventricle is called bicuspid valve. Both tricuspid valves and bicuspid valves are connected to these muscles. They're called papillary muscles through chordae tendineae. Chordae tendineae. These papillary muscles, when they contract, they pull the valves, they pull these strings and open the valves. So the blood can stream from the atria into the ventricles. There is a wall in between the two ventricles. It's called interventricular septum. We have the same wall, which is not shown here, between the two atria. It's called interatrial septum. Here is showing us ascending aorta. At the very opening of the aorta to the left ventricle, we have a valve. This is called aortic semilunar valve. Aortic semilunar valve. We have a pulmonary semilunar valve, but it's not shown here. Here, again, we can see the pulmonary semilunar valve. This is pulmonary trunk. This is ascending aorta here. And this is superior vena cava. This one is your right atria, left atria, a little bit of it you can see. Left ventricle, right ventricle, tricuspid valve, bicuspid valve. You have chordae tendineae on both sides. You have layers of the heart from outside in. Is called epicardium. This layer, the meaty part of it, is called myocardium, and the inside layer is called endocardium. We're looking at the anterior view of the heart, endocardium. Here again, we're continuing to look at more structures. This is a coronal section of the heart, and it's showing us the anterior view. I can see the right auricle, left auricle, right ventricle, left ventricle, tricuspid valve, chordae tendineae, bicuspid valve. We have papillary muscles shown here. We have this band, which only is seen on the right side. This is called moderator band. Moderator band is there because the outer layer muscle of the right side is actually very skinny, and that moderator band, when it gets too distended with the blood, it can bring the wall back to its normal size. How do I know where my right side is after I have a coronal section? Which section is showing me the anterior view on the right side and on the left side? The outer wall of the left ventricle is much thicker than the outer wall of right ventricle. Reason being because left ventricle has to pump blood to the whole entire body, but right ventricle is just going to the next door neighbor to the lungs. So that's why we also have moderator band only on the right side. Here we're looking at the right coronary artery. These are the arteries and veins that supply the heart muscle themselves. So we have right coronary arteries, left coronary arteries, which we see very little of it, and then it will be branching into circumflex artery and anterior interventricular artery. Here we also, on the anterior view of the heart, we also see great cardiac vein, great cardiac vein. If you look in the posterior end, we see posterior interventricular sulcus, and we see coronary sinus. Coronary sinus is a big chunk of blood vessel in the posterior end. It's a blue blood vessel. You have to look in the posterior end, coronary sinus. We also have azygous vein, this one vein. This is not the coronary part of the coronary circulation, but it is located in the posterior end, and it actually feeds into the superior vena cava. Okay, here we're looking at the heart dissection model. It's already coronary sectioned, basically from the apex of the heart, from the tip of the heart. You make the blade and you make a straight section all the way to make two complete halves. Here I'm looking at, in this half, I'm looking at left atrium, right atrium. I have tricuspid valve, bicuspid valve, chordae tendineae, papillary muscles. We have the anterior interventricular septum, aortic semilunar valve we also can see here. Bicuspid, aortic semilunar valve. And then here is showing us the apex, the tip of the heart, and the base. Here showing us the layers of the heart. Epicardium, myocardium, and endocardium, which is inside layer of the heart, endocardium. Here we are looking at the coronal section in here. When I'm looking in here, it's showing us aortic semilunar valve, also showing us the epicardium, myocardium, and endocardium layers in the sheep heart. Here in the posterior section, we have posterior interventricular sulcus, which is going straight up. And it actually goes straight up to, you could see inferior vena cava, this collapsed vein on the top right here, which if I poked it with my probe, it would take me to the superior vena cava, which I don't have it here to show. This shows right auricle, and this one's your left auricle. So right ventricle, left ventricle. Here, we're going to move towards the blood vessels. Here, I'm seeing cardiovascular system. So I'm seeing part of the heart and part of the blood vessels. Again, to orient ourselves, this is anterior view. You have right ventricle, left ventricle, right auricle, and left auricle. This is pulmonary trunk, which is branched into pulmonary arteries in the right and left, and pulmonary veins on the right and left in red. Pulmonary arteries are blue. The reason being because they're bringing deoxygenated blood to the lungs. So whatever leaves the heart is artery. Whatever comes back to the heart is vein. So the oxygenated blood that comes back from the lungs is vein. Whatever comes back to the heart is vein. Here showing us ascending aorta and aortic arch. And this is pulmonary trunk. Between these two, there is a ligament connection. It's called ligamentum arteriosum. This ligamentum arteriosum is actually an opening in fetus. So circulation of the blood is faster. And then after the baby comes out of the birth canal, this, with the first breath, this will be filled with ligaments and it will be closed and becomes ligamentum arteriosum. Branches of aorta is brachiocephalic artery here, left common carotid artery, left subclavian artery is this one right here, that's not marked. We have superior vena cava branched into left brachiocephalic vein and right brachiocephalic vein. Here again, we see great cardiac vein and we see anterior interventricular artery. Red is the artery, blue is the vein. They're sitting inside the anterior interventricular sulcus between the two ventricles. Here we're looking at the lateral view of vascular system. This is left auricle, left ventricle here, and I see a part of chordae tendineae here. Pulmonary trunk will branch into pulmonary arteries on the left side and on the right side. We see a beautiful ligamentum arteriosum. The spelling is incorrect. Ligamentum, E-N-N, we have to correct it. Aortic arch, pulmonary arteries, pulmonary veins. These two are pulmonary veins. you have ascending aorta, aortic arch, thoracic aorta in red. You have superior vena cava branched into brachiocephalic veins on the left side and on the right side. You have brachiocephalic artery right here, left common carotid artery, left subclavian artery. These are branches of aorta. I can see esophagus here, and this is trachea right here. This is thoracic aorta. Here we're looking at major arteries and veins. So we have, from here, we have the brachiocephalic artery is divided into subclavian artery and right common carotid artery. Right subclavian artery becomes axillary artery under the arm right here. Then brachial artery on the arm will branch into ulnar artery and radial artery. And then for the legs, you start with, these are common iliac veins, becomes internal iliac veins, external iliac veins. As far as the artery goes, common iliac arteries. And then you're going to have internal iliac arteries, which is not shown, and external iliac arteries. It will go, becomes femoral artery, popliteal artery, and then anterior tibial artery comes to the front. This is the anterior end of the body, and this is the pulmonary trunk right here. Here we're going to look at, so the heart location is here. Superior vena cava is branched into right brachiocephalic vein and left brachiocephalic vein. It's further branched into right internal jugular vein and left internal jugular vein. You also see here right common carotid artery on both sides, right and left. And here, greater omentum is this connective tissue sheet. It's a covering that holds this internal structure of abdomen in place. It's called greater omentum, part of the digestive system. This is looking at the anterior view of the major arteries and veins, and the greater omentum was there, so I just decided to show it to you. Here, we're looking at the common carotid artery further branching into external carotid artery and internal carotid artery. Internal carotid artery goes to the brain, external comes to the face. And you have it on the right side, you have it on the left side. Usually when you have common of some sort of artery or vein, it will divide into internal and external. We're going to continue with the major arteries and vein. You have right ventricle, left ventricle, just so you know we are on the anterior end, apex, base, right auricle, left auricle. This one is ascending aorta, aortic arch, and then it goes down to descending aorta or thoracic aorta. Above diaphragm, below diaphragm is called abdominal aorta. You have branches of aorta. Here also brachiocephalic artery further subdivides into right common carotid artery and right subclavian artery. Then you have left common carotid artery, and in the very back, it shows a little bit of left subclavian artery. Superior vena cava is further divided into right brachiocephalic vein and left brachiocephalic vein. They are divided into right subclavian veins and right internal jugular veins and right external jugular veins, which we don't see it here. Then with the veins is the same, that we're going to go to axillary veins and axillary arteries. We go to brachial veins and arteries. Then we go to ulnar veins and radial veins. Here it's the anterior view. We're going to further explore that the common iliac arteries at the ilium level on the right and left side will branch into internal iliac arteries in red or into iliac veins in blue, and external iliac arteries and iliac veins. So iliac arteries are in red and iliac veins are in blue, blue. Here we are looking at three famous branches of abdominal aorta. I wrote CSI, like a sitcom in a TV show, celiac trunk, superior mesenteric artery, and inferior mesenteric artery. They're actually located on the, or branched from the abdominal aorta, starting from celiac trunk on the top, then superior mesenteric artery, and then inferior mesenteric artery. These are located on the anterior view of the trunk. Gonadal arteries and veins, they are very stringy-like structures that you can see here. This is, again, our abdominal aorta. It shows, again, celiac trunk, superior mesenteric artery, and inferior mesenteric artery. It shows us the gonadal arteries in red, these stringy structures, and gonadal veins in blue on both sides. Again, these are common iliac arteries, which then become internal and external iliac arteries. Here I'm looking at formed elements of blood. These are the cells of the blood. Erythrocytes, or red blood cells, you have tons of them. They don't have nucleus. They look like life saviors. These are important for carrying oxygen, bringing oxygen and nutrients to the different organs. But your white blood cells, which are all of the other ones, these are very few in numbers, very few. Like in one section of the slide, you might see one or max two. And then the rest is just the red blood cells, the erythrocytes. So you have basophils that they have dark blue granules. You have neutrophils that are multinucleated. You have eosinophils that have red granules, and you have lymphocytes that have a very large nucleus. They're larger, and they have a huge nuclei. And then monocytes, usually you have bean-shaped like nucleus. Okay, so these are the nucleuses inside of these cells. This whole entire thing in lymphocyte is the nucleus, and this bean shape is the nucleus. Erythrocytes have lost their nucleus, so you have enough place to carry enough space. Here it shows platelets. Platelets are really not cells, they are cell particles, and that's how they look like. These are circulation to the brain. It's called circle of Willis. From this black arteries, black color arteries, is called vertebral arteries. They merge to become basilar artery. Basilar artery feeds into posterior communicating arteries, posterior cerebral arteries. These are internal carotid arteries, these two. They go to middle cerebral arteries here, and anterior communicating arteries and anterior cerebral arteries. This is the posterior end of the brain, and this is the circle of Willis. Hepatic portal circulation shunts blood from the digestive organs to the liver for further metabolism and to detoxifying. And from liver through the inferior vena cava goes back to the heart. So this is model of liver. I am looking at the inferior view. This is inferior vena cava. This is hepatic artery, and I'm looking at hepatic portal vein. I don't really have the whole hepatic portal circulation here, but hepatic portal vein is shown in purple. Your hepatic portal circulation has superior mesenteric artery, inferior mesenteric artery. These are bringing the blood from the intestines. You have splenic artery, and together they merge to hepatic portal vein to get into the liver. This is gallbladder. Here you're looking at lymphatic system. This is related to the body's defense mechanism. We are looking at these little dots. They are lymph nodes, and depending where they are, the name comes from it. Inguinal lymph nodes, these are under the arm, so they're axillary lymph nodes. And this passageway, this duct, is called thoracic duct. It's in the thoracic area. And cisternochyle, this bulged out structure, is a pooling place from all these lymph nodes from the lower extremities. They come here, they get concentrated, and through the thoracic duct, they go and they merge to left lymphatic duct and right lymphatic duct. Left lymphatic duct sometimes is called thoracic duct as well. Here, these are cervical lymph nodes. These are axillary lymph nodes, and you see the right lymphatic duct and left lymphatic duct. This is showing an electrocardiogram here. This is the recording of heart electrical activity. You have different waves, different peaks. Each of them have a name, and each of them occur after a certain event in the heart. For example, you have P wave, QRS complex, which has Q wave, R wave, S wave. Together, it's called QRS complex. You have T wave that is coming after QRS wave. You have on the y-axis, you have the amplitude of response in millivolt, and on the x-axis, you have time in seconds. P wave corresponds to atrial depolarization. This occurs before atrium contracts. And this is generated by sinoarterial node or SA node. Then you have QRS complex, which corresponds to ventricular depolarization, which occurs before ventricular contraction. Ventricular contraction is called systole and occurs somewhere here. So we're looking at the electrical activity, and what follows is the contraction or relaxation of the muscle. So here you have in the T wave, you have ventricular repolarization, right? In QRS complex, we had depolarization. Here is repolarization, electrical event. Then this one follows by ventricular relaxation, which is called diastole. So you have systole, ventricular contraction, and diastole, ventricular relaxation. In the lab, we do this blood typing that we use these basically blood typing slides that have these cavities, these depressions in them. You put 2 drops of your blood in question, and we use synthetic blood in the lab. And then we use anti-serum against the antigens that cover the surface of the red blood cells. You have antigen A, antigen B, and antigen RH. So we're going to put anti-serum against those antigens. You put anti-B in one well, which is marked with B, anti-A in one well that is marked A, and anti-RH in the well that it says RH. Then you put the slide in a light box and observe the well for clumping. If the antigen in question is present on the surface of the red blood cell for that subject, you will see the antigen and antibody find each other and clump. The correct terminology is agglutination. Agglutination means they find each other, they clump, and they precipitate. So you see a precipitation on that well. So that tells you that antigen is present at the surface of that subject, the blood of that subject. And I'm going to have some examples here for you. For example, if we are looking at agglutination in B well and nothing in A, nothing in RH, we call that blood type B minus, because minus or plus shows if the antigen Rh is present in the Rh well or not. O plus means there is nothing in A, nothing in B, but you see agglutination in Rh well. AB plus means you have agglutination in all three wells, A, B, and Rh wells. Here we're looking at blood typing. How do we blood type a subject? Basically, you have antiserum A or antiserum B or antiserum Rh, and we're looking at the agglutination amount. And if you have agglutination, that means that antigen is present on the surface of that red blood cell that we are looking for blood typing. Otherwise, if there is no agglutination occurring in the well after you mix the antigen and antibody with each other, you have no antigen present at the surface. So anti-A serum, if you have agglutination, A antigen is present on the blood surface. Otherwise, no antigen is present. Anti-B antiserum added to the wells, agglutination occurs, B antigen is present on the surface of blood cells. Otherwise, no B is present. Same with anti-Rh antiserum added to the blood in question. If agglutination occurs, that means antibody found the antigen. That means Rh is present at the surface of these red blood cells. And if there's no agglutination and it's like a clear liquid, then there's no Rh antigen present. And Rh only tells you if it's plus or minus. So A plus, A positive, or A negative. This is upper respiratory system. This is mid-sagittal view. We have external nares. We go inside the nose and we have these bones that they're called superior conchae, middle conchae, and inferior conchae. Then we go to the back of the nose. It's called internal nares. And we're going to go through here to this passageway. It's called trachea. And from trachea, we go to the lungs. This part, pharynx, is shared with both respiratory and digestive system. Here we are also seeing the frontal sinus and sphenoid sinus, these spaces that are important for the speech also, for the sound that comes out of our mouth. Here from this area, trachea, we're coming down to get to the lungs, but right now we're going to cover this beginning of trachea. So we're right here. It's called larynx, this beginning. It's called larynx, and it starts with thyroid cartilage, and all the way to cricoid cartilage it ends. So this is, and then you have hyoid bone here also. Hyoid bone, thyroid cartilage, and cricoid cartilage, they make larynx. You come down to trachea. Trachea branches into primary bronchi, secondary bronchi, and tertiary bronchi. You have upper right lobe, middle right lobe, and lower right lobe of right lungs, upper left lobe, and lower left lobe of left lung. So on the lung, on the left side, you only have two lobes because of the location of the heart, which also there's an indentation here. It's called cardiac notch. That's where the heart is located. This is the base of the lungs, and these are the apex of the lungs. You have two fissures. One is called horizontal fissure on the right side and right oblique fissure. On the left side, you only have this left oblique fissure. Fissure is kind of like a very shallow or actually very deep groove. So left oblique fissure, right oblique fissure, horizontal fissure, only on the right side. If we are going to take this larynx and we're going to make a diagram, it's going to show us the hyoid bone on the top, thyroid cartilage, and cricoid cartilage. All of these together make larynx. From here, we go to trachea. We also see thyroid gland here, and we also see epiglottis, which closes when we're eating. It closes this passageway so the food doesn't go to the lungs. Spirometry is a technique that we use to measure respiratory volume, measure lungs function. And this is basically the device that we use. It's called a spirometer. And in the lab, you have to do the experiment and you have to look at this respiratory volume and different variables in the respiratory volume. We switching gears to the digestive system. We have the oral cavity. Oral cavity takes us to esophagus. From there, we go to the stomach and we go to the small intestine and large intestine. And we have supporting organs like spleen and liver and pancreas that is not shown here. Here again, esophagus takes us to the stomach. We have spleen here, gallbladder. We have the duodenum at the very beginning of the small intestine. It's called duodenum. We move to be named in the small intestine jejunum, which is most of the small intestine. At the very end is called ileum, which there is a valve here. It's called the ileocecal valve, which is a connection between small intestine and large intestine. So cecum and ileum, they have a valve in between. It's called ileocecal valve. Cecum is the beginning of colon or large intestine. You have ascending colon, you have transverse colon, and you have descending colon on the other side. It gets to the sigmoid colon and rectum and anus. You have two flexures, two bends. One is called right colic flexure, and one is called left colic flexure. Here again, just to show you the greater omentum, which is a covering that covers these internal structures in the abdominal area. Okay, here we have the sagittal head that we saw, but this is a mid-sagittal section. We're only focusing on the digestive portion of this. We have the opening of this is the mouth, the opening of the digestive system. We have tongue. Here is the hard palate, soft palate, and the end of it, uvula. We have these tonsils. This one's called lingual tonsil. You have palatine tonsils, and you have palatine tonsils, and you have pharyngeal tonsils. This is the opening of eustachian tube. Is not necessarily part of the digestive system, but I just wanted to show you. It's called opening of eustachian tube in the ear. So it goes to the throat from here. This is the tongue, oral cavity. Pharynx, this passageway is shared between the both respiratory and digestive system. It has sections to it. You have nasopharynx where the nose area is, oropharynx in blue or in purple where the mouth is, and laryngopharynx when you're getting to the larynx area, laryngopharynx in green. This is a mid-sagittal section of the head. Here is showing us the salivary gland. This is the tongue. This is the parotid gland by the ears. Submandibular gland is by the mandible, and sublingual gland is under the tongue, and it shows the duct of these glands as well. Okay, anatomy of the stomach. Again, from esophagus, we're going to come to the stomach. From stomach, we're going to go to the duodenum. But we're looking at the stomach. The region on the top, the bulged out region, is called fundus. The rest is the body of stomach. You have these folds, rugae, that allow for the expansion of the stomach when you eat a meal. There is a sphincter here between the stomach and the duodenum. It's called pyloric sphincter. the region by its side is called pyloric region. You have two bends or two curvatures, lesser curvature and greater curvature. You have these muscularis layer that are on top of each other. The outermost is called longitudinal, circularis, and oblique. These are layers of muscles on the stomach. You have another sphincter here between the esophagus and the cardiac region of the stomach. It's called cardioesophageal sphincter. Cardioesophageal sphincter. Here we're looking at the small and large intestine, the wall of them. This is how you can see it. You have the outermost layer of this organ system. It's called serosa. You're going to go in to the longitudinal, then you go to circular, then you go to submucosa, and then the very inner layer is called mucosa. So this is the lumen where the food goes through. So you go mucosa, submucosa, circularis, and longitudinal. And the very top layer covering the longitudinal towards the outside is called serosa. So this is the small intestine, and this is the large intestine, and this is the stomach. We're looking at the liver. This is the supporting organ to the digestive system. This is the superior posterior view. You have left lobe, which is smaller because of the location of the heart, and right lobe, and they are divided from each other through this falciform ligament. Here we are looking at the inferior view of the liver, and it's showing us the bile duct system in green. So you have gallbladder, cystic duct from gallbladder, common hepatic duct from the liver. Together, they merge, they become common bile duct. So again, cystic duct from the gallbladder and common hepatic duct in green from liver merge to become common bile duct. Here we also look at hepatic portal vein. We saw it before, hepatic artery. We have inferior vena cava again, and we are looking at quadrate lobe here, quadrate lobe, and caudate lobe here. This is caudate lobe. There are two ligaments. One is in white, round ligament of the liver, and ligamentum venosum. Here we're looking at stomach, pancreas, duodenum, and gallbladder. So here we look at the bile duct again in another close-up look. We have cystic duct, common hepatic duct from the liver here, merge to become common bile duct. Common bile duct will empty its content into duodenum, the beginning of small intestine, through this opening, which is called duodenal papilla. Pancreas also does the same thing. The pancreatic duct also enters its contents, its juice, inside the duodenum through duodenal papilla. Here we are seeing the pancreas in yellow, duodenum in pink, and gallbladder in green. Duodenal papilla is also seen here, and this part of the pancreas is called head of pancreas. This is the body of the pancreas, and at the very end, we have the tail of the pancreas. And these are the pancreatic ducts. We have pancreatic artery and vein also. Urinary tract, we're going to review the structures in the kidney, and we're going to look at nephrons. Nephrons are the functional units of kidney. There are hundreds and thousands of them. We're going to just magnify one, and we're going to look at that one. When we get to know about renal corpuscle, I want you to pay attention. There are two arterioles, one's supplying and one's draining this corpuscle, renal corpuscle. And the thicker one is called afferent. The thinner one is called efferent arteriole. Okay, here I'm looking at the anterior view of the urinary tract. These two are kidneys. I have adrenal glands above the kidneys. Kidneys are covered in these capsules. They're called renal capsules. This point of entrance of these tubules and these arteries and vein is called renal hilum or renal hilus. You have from the kidney, you get to ureter, and from ureter, you go to the urinary bladder and exiting the urine from the body. Here we're again looking at abdominal aorta. And if you remember, we had CSI, we had the celiac trunk, superior mesenteric artery, and inferior mesenteric artery here. We have at the ileum level, we have the common iliac artery and vein, artery in red, vein in blue, and they become internal and external iliac arteries in red and veins in blue. Okay, so we're going to take cross-section of kidney and we magnify. This is our anterior view, and we're looking at the internal structure. So the renal capsule is the capsule that's covering the kidney. This layer is called renal cortex. And this entire layer inside the renal cortex, this entire layer is called renal medulla. Renal medulla. You have inside the renal medulla, you have these columns, these areas, they're called renal columns in between renal pyramids. These are renal pyramids, and these areas in between is called renal column. The urine is dripping from these renal pyramids inside minor calyces, from minor, and these are all minor calyces, minor calyce, minor, all of these. Okay, so from minor calyces, they are collected, a few minor calyces collected into major calyces, and all major calyces, they all drain into renal pelvis, and from there it goes to ureter to get out of the body. Here again, I'm looking at the cross-section of kidney. You have renal column, renal pyramids, renal cortex, renal medulla inside, again showing us minor calyces. Together they make major calyces. From there we get to the renal pelvis. From there we get to the ureter. And renal hilum is the point of entry of these blood vessels, renal artery, renal vein, and renal pelvis. It's called renal hilum. Now I'm going to make a cut here, a pie here, and I magnify it here to look at the nephrons. Nephron is unit, the functional unit, the smallest functional unit of kidney. So if I took a pie and I magnified it here, all right, we're looking at renal corpuscle, this structure, proximal convoluted tubule, loop of Henle, distal convoluted tubule, which goes to collecting tubule. So a nephron starts from renal corpuscle and ends at before you get to the collect tubule. Okay, so renal corpuscle, proximal convoluted tubule, loop of Henle, and distal convoluted tubule. That's a nephron. Here we are also, this capillary bed is called vasa recta. And this capillary bed is called peritubular capillaries. So I'm going to take renal corpuscle and I'm going to magnify it to study it further. I have glomerulus, which is covered by this capsule, which is called Bowman's capsule. Now we're looking at proximal convoluted tubule and distal convoluted tubule. We are looking at afferent, which is the thicker arteriole, and bringing blood to or feeding the renal corpuscle, and efferent arteriole, which is the thinner one. It's draining the corpuscle. As you can see, the proximal convoluted tubule is connected. So if this was renal corpuscle, proximal convoluted tubule is connected. Distal goes down. You actually, from proximal, you go down to loop of Henle and then come back to the distal, which is very close to the renal corpuscle as far as proximity goes, but it's really far because we went long distance to get to it. That's why it's called distal convoluted tubule. This is showing structures in the urinary bladder. You have these folds, again, allowing for the fluctuation of liquid fluid inside. It's called rouillé, like the way that we had in the stomach. You have this triangular structure here at the point of entry of the urine inside this passageway. This is called trigon. This is a male model because we see prostate gland, and this passageway is called prostatic urethra. And this is urethral orifice where the urine empties the body. We're switching to male reproductive system. We have testes. Testes is covered with scrotum. Inside the testes, we have epididymis, and through vas deferens. And this is ejaculatory duct. Through vas deferens, the semen will come through this passageway. And this is our urinary bladder. This is urinary bladder. This is our prostate gland. You have prostatic urethra, membranous urethra at the pubic symphysis, layer. This layer is called membranous urethra, and penile urethra. And then urine and semen exits through this orifice. For the penis, you have the shaft of penis and prepuce, or glans penis, which is covered with prepuce. So shaft of penis, glans penis, which is covered with prepuce. Seminal vesicle. Here you have rectum and anus. Here, anterior view of male reproductive system showing the testes and the penis. However, I want to show you the erectile bodies, these muscular structure. The outer one is called corpus cavernosum, and the inner one, which is more to the medial end, is called corpus spongiosum. We are looking at female reproductive system. Here, again, we're looking at mid-sagittal view. We have, this is called labia mons pubis, right here. This is called labia majora, and you have urinary bladder. This is urethra. We have uterus that has layers. The outer layer is perimetrium. The middle layer is myometrium. The inner layer is endometrium. This is cervix, right here, cervix. You have vagina and vaginal orifice. This is urethral orifice here. This is rectum and anus. And before rectum, you have sigmoid colon. Here I'm looking at ovary and ovarian ligament here, ovarian ligament. Here we could see the round ligament of uterus. This is uterus again, urinary bladder, sigmoid colon, rectum, anus. So this white structure shows round ligament of uterus. Here we're looking at the anterior view of uterus and the ovaries and the fallopian tube, this tube. So the uterus, the bulgal structure is called fundus of uterus, and this area is called the body of the uterus. This is cervix, and this is vagina. You have the ovarian ligaments right here. You have the ovaries, and you have fallopian tube. Fallopian tube is divided into sections. Isthmus is the connection of the fallopian tube to the uterus. Then you have the ampulla, you have the infundibulum, and you have the fimbriae, which is the finger-like projections. Here I'm looking at the cross-section of ovaries, and it's showing me that every month in a menstruating girl, you have primordial follicles. They are maturing to become primary follicles, secondary follicles, Graafian follicles, or ovulating follicles, and ruptured follicle, where the ovum will leave the body. And if pregnancy occurs, the follicle, the remaining follicle, which is called corpus luteum, stays active to support secreting the hormones to support a pregnancy. If pregnancy doesn't occur, then corpus luteum becomes inactive, and it will be called corpus albicans. Corpus albicans. This is the ovarian ligament, by the way. And this is the germinal epithelium. Germinal epithelium. Here again, we look at the follicle development in human ovaries. These are the phases, the ovarian phases of menstrual cycle. You have the ovum inside primary, secondary follicle, Graafian or ovulating follicle. Ovulation occurs. Ruptured follicle here. Corpus luteum. If no pregnancy occurs, corpus albicans. Human breast, we look at skin, adipose connective tissue, pectoralis major muscle from second unit of study, pectoralis major. And then we have areola around the nipple of the breast. And we have lactiferous ducts that bring the milk from these mammary glands. These are lobes of mammary glands. From here, the lactiferous ducts bring the milk towards the nipple, towards the exit. And when they get to the nipple, they become wider. They're called lactiferous sinus. So lactiferous ducts, they become wider, and they are named lactiferous sinus at the nipple area.